Welcome everyone to the next iteration of the IITE seminar series. So today it is my great pleasure to introduce Will Godsoe from Lincoln University from New Zealand. So he's gotten up very early just for our sake today to talk about biodiversity and information theory. Namely, it can be really difficult sometimes to connect the metrics we use to measure diversity with actual biological processes. And that is what Will is going to be talking to us today. So without further ado, the floor is yours. Thank you.
Okay, and thank you very much. And this was mentioned before, and I'm just going to highlight it as well. I'm happy to have questions, but it's 6 o'clock in the morning here. I'm in Lincoln University and live in Christchurch in New Zealand. And so please have questions, but expect me to be a little bit slower than you might otherwise believe. And to that end, I have just noted that I opened the wrong presentation. So give me 1 second. I have one here. Theoretical ecology seminar. There we go. So I pulled this up right now. So this is me, William Godsoe. I'm at Lincoln University in New Zealand. I volunteered to do this presentation in part because it's really nice to talk with people doing theory in other places of the world. There's some very interesting theory that's done in New Zealand, but we are, very much on sort of the far end of the world, and it's really good to have a chance to share ideas around.
Sorry, just one. Sorry for interrupting. Just that right now you're sharing your notes and next slide
yeah. Thank you very much.
No.
That sounds good. Okay, stop sharing that. And share screen 2.
Yes, that looks great.
That is looking much more promising, and that looks like the right title slide.
Yes.
Great.
Thank you.
Okay, so what I'm going to do today is go through some ideas in 3 recent papers of mine. And this is the first time I've done a seminar in this format, so I'm not sure if we'll get through all of the papers, but we can at least start through them. And if there are further ideas, I can refer you to the papers. And of course, if there's things that are unclear, it's possible that things will be clearer in the papers where it doesn't depend on me being completely awake. I'm an ecologist. I'm very much interested in biological diversity. I live in New Zealand, which means that diversity is important, as it can be in lots of other places. This is a photograph from a hut off the west coast of New Zealand, where there's sort of cool facets of diversity, including you could hear kiwis singing off in the distance and on this landscape, and then rare short seabirds going out to hang out at sea over here. So I know that I am one of many people who are interested in diversity. There's sort of a broad sense in ecology that diversity is a very important part of what we do. It's important for things like productivity. It's interesting to lots of us. But we're often in the situation where changes in diversity can be hard to predict. And so, if we think about diversity in terms of individual species like kiwi, we have ideas for how to make models for it. But just oftentimes, long-term trends in diversity can be surprising or unintuitive or difficult to sort out. So I put up here a photo of a paper in Trends in Ecology and Evolution from 2016 called How Should Beta Diversity Inform Biodiversity Conservation? And basically, the paper says beta diversity, which is an important way to measure diversity, is something that we should think about in conservation. There are a bunch of mechanisms that can change beta diversity. And each of the mechanisms we can think of sometimes increase beta diversity and sometimes decrease beta diversity. And so because of that, there's lots of things that we think are important, but we don't always know what they do. All right. A while ago, I was invited to an IDIV working group where I was a very small part of a review paper on improving forecasts of beta diversity change. Improving forecasts for biodiversity under climate change. And the working group was amazing. I learned a lot. I contributed a small amount to the paper, including sneaking in a photo of myself in one of the figures. But much more of it is stuff from Mark Urban and a number of the other lead authors on the paper. But in the paper, one of the things that we talked about was sort of listing mechanisms that we thought were important for measuring diversity and trying to integrate those mechanisms into our predictions for diversity in the future. And to me, I think this is sort of a useful snapshot for how we tend to think about what's happening. So we have at the top a mathematical model that we define, which is sort of a description about how communities work over time. We can think about things that we want to put in the model. We can then obtain projections based on that model. We can get more data and then just go through this beautiful cycle of refining our model. And I think that's how we often try to sort out this type of problem. But looking at this review and looking at other stuff, we just know it's extraordinarily difficult. It's hard to get information on all the pieces that we think matter in ecology. And so in this talk, I'm going to put together a couple of ideas that I've had in papers of mine to try to come up with another way to think about our models of diversity change. And to do this, what I'm going to pose as the idea is that the challenge we have when we're modeling changes in biodiversity is sort of a matter of integrating several hard problems at the same time. So one of the problems is just, how do we measure biodiversity? What does it mean? At the same time, we're often stuck with problems of statistical statistical inference. So how do we gather data on the pieces that matter to us? And we're also stuck with problems of coming up with ways of developing dynamic models or mechanistic models where we're describing how things change over time. And the big thing that's driven the research I'm going to present in these papers is we treat these three ideas as really different from each other. So we have like fancy measurements of diversity, fancy dynamic models, and then fancy tools for statistical inference. But there are ways that we can treat these all as a common language. And in particular, one of the ways that we measure biodiversity is using tools from information theory. So we stole a bunch of our ideas for biodiversity measurement from Shannon and other measures of entropy. When we're thinking about statistical inference, a lot of the ideas we have are grounded in information theory. So like a lot of the theory we have about how to do statistical inference properly has an information theoretic interpretation. And at the same time, there are ideas that we can steal from evolution for how things change over time that have an information theoretic interpretation as well. And so instead of thinking about a bunch of very disconnected problems, maybe we can start to squeeze all these problems together into one big happy piece. So over the course of this talk, I put in ideas from 3 of my papers, and I'll start talking through these right now. And if we get through them, great. And if we don't, we can just cut through for 1 or 2 of them. So the first paper talks about how biodiversity can obscure the mechanisms that ecologists would like to study. The next paper then sort of takes this idea of information theory and uses it as a way to rethink the mechanisms that change biodiversity, focusing on effects on the rarity of individual species. And then the final part applies this to look at, to measure the causes of diversity change in a real community, focusing on beta diversity. Okay. Now, I've been talking for a fair bit. Should I just pause? Is this working? Are there any questions before I go further? I should ask you, am I talking at about the right speed?
From my end, absolutely. Usually what we, so I think it's good to, if people have the opportunity to ask questions in the middle of the talk, but usually the way we do this is that we save the questions to the end. That should not rule out people asking, of course, but just don't be surprised if they if they pile up at the end instead.
Okay, great. All right, thank you. Good. Okay, I'll keep plowing along then. So the first results I'm going to present are from a paper that's just coming out in Oikos right now, called Species Interactions and Diversity, a Unified Framework Using Hill Numbers. And so in this paper, I'm going to talk about, for this, I'm going to talk about 3 different parts. So the first part is the idea that biotic interactions, which is the mechanism which has interested me the most and which I think interests lots of community ecologists the most. So biotic interactions and diversity have different units. And using traditional methods, it's hard to picture the relationship between the units of the two, but we can develop graphical methods to link the units associated with the two problems. When we link these units at the same time, we can show how biotic interactions, how different biotic interactions can have identical effects in biodiversity. And so it isn't just a matter of we can go out in nature and look at measurements of biodiversity and tease apart which interactions matter. Instead, because of the way the units work, many different biotic interactions can have similar effects. So to start off, I'll just say that the community ecology has long been concerned with interactions among species. There are other mechanisms, but this is a common one, and it's the one that I pitched this paper around. So by interactions among species, I mean basically that we have multiple species that are out in nature, and each species can have an effect on the growth rate of other species. One way that this is summarized is using what's called the wheel of interaction. It's the idea that you can have things like a mutualism where both species benefit for each other, competition where both species harm each other. We can have predation or prey and all these different types of interactions. And then, so then this is one simple way to picture biotic interactions. It's not the only one, but it's a really handy starting point. The way that our mathematical models work, we tend to define biotic interactions based on their effects on absolute abundances of species. So what I mean by this is that if we write out a model for biotic interactions, either a simple model like this is sort of like a generalization of a Lotka-Volterra model, or a consumer resource model, or whatever it may be, the units that we're interested in concern the absolute abundances of the species that we're working on. So here, for example, I have some lovely fruit flies at the bottom of the picture, and I can count these 1, 2, 3, 4, 1, 2. And then the mechanisms that we define change those absolute abundances of our species. So I've written the model here. So we've got this part of the model, which works like logistic growth. And basically it says species grows by itself, and then it reaches carrying capacity. And then this part over here, which defines the effects of one species on another. This is not how we usually count when we measure diversity. When we measure diversity, what we're usually doing is counting relative abundances. So for example, with this fruit fly example here, we'd say that the probability that a fruit fly belongs to the orange species is 2 out of 3, and the probability it's blue is 1 out of 3. Just to drive that home, you can see that in diversity indices and true diversities, or many of the different ways that we focus on diversity. I've put 2 of these up here. In New Zealand, many people prefer to work on classic diversity indices. And so I'm just touching on this part here, but we can discuss this one as well if more details are needed. But basically, if you look at Shannon entropy, which is a measure that's familiar to most of us, you've got a PI. So this is a relative abundance, and then another PI over here. We'll get to why in the future, but it focuses on relative abundances. And when you get to fancy contemporary diversity measures, like a Hill number here, this is a Hill number is a generalization of lots of different diversity indices. And again, we've got a PI over here, which is the information you're getting from the community. And then you've got a Q, which tells you how much to focus on rare species versus common. But in either case, we've got units of absolute abundances for biotic interactions and units of relative abundances for diversity. This fact means that we can miss a bunch of information when we analyze diversity. So for example, here I show a community where the abundances of fruit flies have declined over time. So in the past, there were 6 fruit flies. And then in the present, there are 3 fruit flies. Diversity has not changed. Likewise, here we've got a community where the absolute abundances have increased. So there should be a total of 6 individuals in the past, and then 12 in the present, if I've done my math right, and diversity does not change. And likewise, you can have replacement between species. So in the past, you've got, the purple species is the most common, and in the present, the orange one is the most common. And again, diversity does not change. So these unintuitive effects of diversity, like if you put them on a picture and do the counting, it's not too hard to spot them, but it's really hard to intuit these by doing traditional analyses of species interactions. So we can write out a mathematical model or a graphical model of interactions between species, and it's really designed to tell us about what's happening in terms of absolute abundances. So here, for example, we've got a phase portrait. It's a graphical model for absolute abundances. And when you've got that, you can see here the abundance of species 1, abundance of species 2. We have the species that are competing with each other, and they reach an equilibrium up here. But this graphical depiction is only telling us about absolute abundances. And I will highlight one more part here before I forget. The key part of reading this sort of diagram is that you've got these arrows here, which tell you the change in absolute abundances across the graph. So that's a classical phase portrait. To help picture what's going on in this paper that I put out, we've worked on modifying this graphical analysis so that we can picture what's happening in terms of diversity. So I'm taking the same phase portrait here. So we've got the absolute abundance of species 1 and the absolute abundance of species 2. On that, we can see the point where you start. So 4 individuals of one species and 2 individuals of the other species. And then look at the change over time. And so we've got an arrow for 1, which is showing decline in abundance, arrow for 2, which is showing increase in abundance, and arrow for 3 showing replacement. On a graph of absolute abundances, you can see that each one of those represent a change. But it doesn't give you much intuition for what's happening in terms of relative abundances. To fix that, you can redesign the axes on the graph. So in the second graph, I put the same pieces, but I've done it but I've labeled on it the different relative abundances on it. So this line here shows a relative abundance of 50-50. This one here is all the places on the graph where the relative abundance is 4-2. And looking at this, we can now see that in terms of relative abundances, the decline makes no change and the increase makes no change, but the replacement does make a change. So that's the first modification, to take sort of the textbook way that we have models and move to relative abundances. But you can go further than that. You can then take that graph of relative abundances and convert it into a graph of diversity. And that's what I've put in here. So what the shading is highlighting is places where diversity is highest, where both species have a high relative abundance, and then places where diversity gets lower and lower as one species starts to dominate. And on this modified version of the graph, you can see that decline keeps you in the same spot, increase keeps you in the same spot, and then the switch keeps you, and the replacement keeps you in the same spot. And so by modifying the graph like that, you can then suddenly convert from the analysis of absolute abundances that we're used to one in diversity. This is the graphical version. I think it's really helpful. In the paper, we do the full math version of this as well. And I'm not going to, and we can talk about that, but I won't put the equations up for it. And in effect, what I'm doing is going from standard models of population dynamics, like Lotka-Volterra, taking what's called the replicator equation from evolutionary theory to convert that into a model of relative abundances, and then using a bit of calculus to modify that again to get expressions for change in diversity over time. So that's the graphical tricks. Once you do that, you can find spots where different species interactions can produce the same effects on diversity change. So what I've done here is pulled up pictures for a sort of a standard phase portrait where you've got a trajectory here for two species that compete. You have one species that has a carrying capacity of 0.5 over here, another species with a carrying capacity of 0.5 over here. If you left each species by itself, both of them would reach, each would reach carrying capacity. But because they compete, their abundances decline and reach an equilibrium where one species is more common than the other, but they coexist. So that's a competitive interaction. On the other hand, here I've got a mutualistic interaction where the dashed lines show the carrying capacities of the two species by themselves. When you allow the species to interact, both of them benefit, and so then their abundances increase sort of past their carrying capacities. So we've got the graph there for competition and mutualism, and you can look at the change over time in terms of diversity, and you can do it for different ways to measure diversity, and both of the graphs show essentially the same qualitative pattern, where diversity rises and then drops rises in the first part of the graph and then drops over time and stays steady as we approach the point where the species coexist. I then put in an example of how this ambiguity could work in empirical examples, which is great, but I think that I'm going to chit-chat a lot and I'm pretty tired, so I'm going to skim over this right now, but it's in the paper as well. And I'm just going to finish up this section by mentioning that some of the ideas in this paper are distilled really nicely in parts of Twitter that now seem to be sadly gone. So we've shown that competition and mutualism can have similar effects on diversity, which I think was beautifully summarized by Wint, who once said that who had the following tweet many years ago: "The wise man bowed his head solemnly and spoke, 'There's actually zero difference between good and bad things.'" And then there's some profanity that I can't share with the talk. So I'm going to summarize this part here. Biotic interactions change absolute abundances, but diversity depends on relative abundances. Phase portraits can be modified to link biotic interactions and diversity, and these methods make clear that many biotic interactions can produce the same changes in diversity. And I'm going to have a bit of water right now, and I'll pause for a second. How would you guys have any questions? Should I just go on to the next section after this?
If anyone has any questions, please go ahead, of course.
I see Giuseppe.
If not, then you can go ahead.
Yeah.
Or sorry,
Can you hear me?
Yeah, go ahead. Sorry.
Yeah. Thank you for the interesting talk. I just had a very quick question about is the Hill number related to Rarity entropy or so? Because I'm not familiar with that. And how do you choose Q? I see that there is a lot of debate in other fields like spin glass and so on. So
That's so great.
How do you get around?
It is a Rarity entropy. And again, I'm used to different audiences, and so like, I wouldn't tell ecologists that, but yes, so a Hill number is sort of what ecologists have called Rarity entropies, and you can put in different values for Q, which correspond to qualitatively different and which match up with different standard diversity indices. So Q equals 0 gives you something like species richness. Q equals 1 gives you something like Shannon entropy, although it takes some calculus. It's technically the exponential of Shannon entropy. And then Q equals 2 gives you Simpson's diversity. So there's this nice mapping between the Rarity entropies and diversity measures in ecology. And I think that's most of what I'd say right now. The best paper for this is by Lou Jost in 2006 in Oikos. And he explains it better than I could right now. But would you like more detail right now, or is that?
No, that's okay. I was just curious if you could somehow pinpoint the value of Q but probably you would need a topology in the model because here I see that this sort of mixed population where everyone interacts with everyone, where usually, let's say in other contexts, at least I saw that you could relate the value of Q to the long-range interactions between species and so on and so forth. But yeah, thank you.
Yeah. That seems really good. I don't see any other questions popping up, but that's a helpful pause for me. I'll keep going then. Okay, so for the second part of the talk, I'm going to talk about mechanisms that change species. I'm going to argue that mechanisms that change species rarity change biodiversity. So in the Oikos paper that I just went through, there's sort of a very high level description of the mechanisms that change diversity. But even though it's quite helpful, it relies on a fair bit of calculus, which can be a communication barrier to at least empirical ecologists. And in addition to that, I have not come up with convenient ways to analyze all the ideas in that paper empirically. They may be there, but because they have a lot of calculus, they're less convenient than other things we want to do instead. So as a result of that, I'm going to highlight another related approach that I used in a paper in Theoretical Ecology a year or two ago now, that tries to give us a sort of a more precise picture about how the mechanisms through which diversity can change. So to get this picture, what I'm going to do is dig a little bit further into what diversity is and how we measure it. So normally when we present a diversity index, we put up sort of an equation like this. This is the equation for Shannon entropy. We look at it, and I at least have got to say that it took me quite a long time to get intuition for what this equation is actually telling me. And I know I'm not alone in this, although there are resources if you dig far enough into ecological theory to really picture what the equation is doing. And so I'm going to go very slowly through the interpretation that I find most intuitive, because I think it's a natural way to link to the mechanisms that change diversity. So when thinking about a diversity index, it's helpful to me to picture this as a way to think about all the individuals in the community. So when we have diversity, we can think about different individuals. So I've got one tree, a second one. And then one part of the diversity index is a score associated with different individuals in the community. So that's this part here. So this negative log pi has interpretations in other disciplines, and more or less what it corresponds to is a measure of how surprising each individual is. So I've tried to highlight this here. So we've got two species of plants. This one is more common. There are two individuals. This one is less common. The one that is individuals that belong to the less common species have a higher rarity score. Individuals that belong to the more common species have a lower rarity score. So that's what this part here is telling us. And then overall diversity is an average of these rarity scores. So basically what that means is that we get a rarity score for individuals of each species, and then the sum over all species weighted by the relative abundance of each species is a way to get an average across all species. So here I've highlighted this for Shannon entropy. It's true for a number of diversity indices that we have. And even when you dig into more complicated diversity indices, you can often find a piece that looks like this. And so to emphasize the generality, I'm going to call these rarity scores Zi. So individuals of each species have a score, and that score tells you how rare individuals of that species are in the community you're looking at. Once we write an expression like this, we can then just take this and think about how it changes over time. I'll put in a little bit of calculus here for this. So we can think about this like this. So we have change in diversity, and I'm putting this like this. So we've got our average rarity score, and then we're differentiating this with respect to time. When we do that, we can then take this derivative and interpret it as mechanisms that change diversity in a community. And I'm going to show you the answer in a minute, but I'm just going to highlight that this sort of approach of taking an average and then differentiating it, it's not extremely common in ecology, but it's very common in evolutionary theory. And so the terminology that I'm going to take is borrowed from evolutionary theory. I see this as an opportunity, but sometimes it causes confusion because it's a different language. So you take the derivative here. People who want to follow along and do the calculus, you can see that the two things that are multiplying together, which gives you a form of the product rule, which gives you two terms. And for now, I'll just describe the two terms as this. So we have a selection term and we have a transmission term. This is analogous to the price equation in evolutionary theory. The selection term lines up quite nicely with things that I think are intuitive to ecologists. So basically what that is looking at is whether species that are rare increase in abundance relative to species that are common. So if rare species win, then the selection term goes up, and if rare species lose, the selection term goes down. So that part is pretty straightforward. The second term, on the other hand, though, you really have to get pictures to see what it's doing. In effect, the transmission term is describing unintuitive changes in diversity, often due to species replacements. So we can go through the calculus if we feel like it, but for now, I'm just going to highlight sort of a simple cartoon example of what the two processes do. This is a bit reminiscent to what I showed in the previous part of the talk, but I'm putting it here to dig in a little bit deeper. So here we've got an example of a community. We've got two species, a moose, which is rare, and then a squirrel, which is common in the past. And then we switch so that in the present, we've got the moose, which is common, and the squirrel, which is rare. So just by the arithmetic of what's set up, in the past community, a third of the individuals belong to one species in the past, a third of the individuals belong to a species in the present. So diversity is unchanged, but there's clearly been ecological changes in the community. And so to highlight why this has happened, I've taken this cartoon and drawn the size of each individual so that it corresponds to how rare individuals of that species are. So the moose is rare in the past, so it's big. The squirrel is rare in the present, so it's big. The squirrel is common in the past, so it's small. The moose is common in the present, so it's small. So once we see those rarity scores, we can then work through why diversity hasn't changed. So we can get the selection term, which is basically saying that moose, the rare species, produced lots of kids. So that's the selection's effect on, so that's selection on rarity. And then at the same time, the change in this community has been so rapid that the rarity scores of each species have switched over the course of our observation period. So that in the past, moose were moose were rare, and then they became common. And conversely, squirrels were common and they became rare. And so those two sources of change cancel each other out in the community. We can find evidence of this in nature as well. So I got data from sort of an aquatic community that Kate Sirianni, one of the authors, had collected. We have small zooplankton species in small ponds off an island in the east coast of the United States. And you can look at total change in Shannon diversity over here. And when we look at that total change, I'll focus on two time periods. We've got one time period over here where diversity goes up slightly, and one time period here where diversity crashes. And what's notable about these two time periods is that in terms of the individual species that are in the community, you can kind of tell the same story at both periods. So there's one species called Brachionis, which increased in relative abundance in the first gray period, and it increased in relative abundance in the second gray period. So when you just look at the time series and you focus on individual species, it seems like similar things are happening. But when you, these seemingly similar effects on individual species resulted in a dramatic change in diversity in one spot and a negligible change in the other spot. To understand what's happening, it's really helpful to tease apart these mechanisms of selection and transmission. And when we do that, what we find out is that in the second time period, you've had selection for Brachionis, which is a common species, which then decreased diversity. So that's pretty straightforward. But in the first time period, Brachionis replaced other species in the community. So it was common. So it was rare and it switched to common, while the other place was common and it switched to rare. And so that replacement results in a very negative transmission term. So you've had selection in favor of species that are rare, and that's been counteracted by this transmission term, leading to no total change in diversity. And so we can use that to think about the mechanisms that change diversity, and to get a better intuition about what's happening. It's helpful to think of this as stating diversity summarizes individual rarity scores. Selection occurs when rare species increase in relative abundance, and transmission describes unintuitive shifts in rarity. And I think that's sort of a good place to talk about what I've got. I had pulled up a final section of the talk where I take these ideas and then apply them to study changes in beta diversity over time using empirical examples. And that's all well and good, but I feel like I've been talking for about 40 minutes now. That's probably enough to discuss for right now. I think maybe I'll pause unless you guys really want to see the final section, and then just open this up to questions.
Thank you so much. I think it's completely up to you whether you would like to go on or would like to stop here. Either works fine. We can start questions. We can go on.
Yeah. Yeah, I'm not quite awake enough. I think it's good to put up a final picture here, and I will thank the collaborators that I put in for the papers. I would be more than happy to talk to people over email when I'm more awake as well. But I think that's enough talking for now. And if you guys are desperate, I will then go into the intricacies of that, and I'll leave it open for discussion right now. Yeah, thank you very much.
That sounds perfect, and thank you so much. Despite the early hour, maybe with the questions, we'll venture into that third part. But in any event, does anyone have a question? If you do, please raise your hand in the participant list. Okay, so maybe while people are thinking, I have some questions. So one question I had, which I guess goes back to the first half of the talk. So in terms of the effects that one gets under, say, competitive and mutualistic dynamics, is that very contingent on things like initial conditions? And I guess more generally, are there generalities, conditions under which we know that the diversity patterns will be similar versus where they won't be? So are these, should we treat these as just case studies that demonstrate that it's possible to get the same outcomes, or is there more to be said there? Thanks.
Absolutely. That seems like a great question. So then it's sort of like, here I've shown examples where you can get a mutualism and competition giving you the same trajectory, and it looks the same for different ways to measure diversity. So, okay. I'm going to start with what I think is provable in the paper, and then highlight things where I think the problem may be too hard to analyze. So the analysis in the paper focuses on instantaneous changes. So like, I have a community, I measure it at one time point, and then I see what happens when I change to the next time point. And in the paper, when you do the calculus, which I can't quite do right now, but you can show that the same changes in diversity can be compatible with growth across your community and decline across your community. And like, any combination, you can have lots of different combinations of growth and decline that give you the same pattern. And on a short time period, that's really what species interactions do. They're things that, like, change the abundances of your species across the community. And so if you have data for like one short time period, then that should be all you should be able to figure out. And just to get more intuition on this, so I put two species in a community. The two species compete. Their interactions are minus. So both species decline in abundance. That leaves diversity unchanged. The two species benefit each other, so they both grow, and that leaves diversity unchanged. And so then if you give me a given pattern of diversity, I can make that compatible with either increase or decrease over a short period of time. Over a longer period of time, it's harder to see what will happen. And the reason for that is that it's hard to explore all the different qualitative dynamics you can get from models of interactions among species. There are many different models. There's chaotic dynamics, there's mess, there's all sorts of nonlinearities and things. And it's much less clear to me that we'd be able to come up with an answer, just because, like, every combination of parameters may be a different set of mess. And so that I'm not sure of, but what I do know is that it's much more common in ecology to have data sets that are short term than it is to have like big, long, elegant, long-term data sets.
Fair enough, and thanks.
Yeah.
John has a question though.
Yeah, this is great. I really appreciate you doing this so early in the morning for you. My question is more about the second part, and I really like the sort of the elegance of the price equation, thinking about changes in rarity through time. And I'm wondering if you've done any sort of more brute force comparisons with some of the other tools that we all use but don't love, for things like temporal beta diversity, some of the turnover metrics, and whether your tool captures those better than some of the uglier tools that we currently use but don't love.
That's a great question, and the short answer is that no, and you've used words which I think are a great way to explain that, which is that sort of, I'm at a small university where there aren't that many people who want to do computational stuff like this, and so I tend to rely on a bit of elegance instead of brute force methods. I think that there are good things that I think it's going to compare well, but yeah, no, I haven't. So for temporal beta diversity, it's my belief that this is better because it gives you direction, right? So like, instead of just saying, oh, there's differences between time periods, what this is saying is it's like, this is how it's declined and this is how it's gone up. And in the part of the talk that I didn't show you from AmNat, I'll just show some of the stuff that you could do. Which is, we had data where sort of like a colleague of mine, Peter Bellingham, who's just up the road, had collected data from long-term forest plots in the Blue Mountains of Jamaica. And a hurricane had come through the site, and you could look at change in diversity. For Shannon entropy, you could take the price equation and pull that out into effects of transmission and selection for each time period, and like tease apart total change in diversity into each of those pieces. So I think there's good there, but by myself, I haven't had the resources to go through all the other tools that are there, which I have I think the same ambivalence that you described.
Yeah, I just Mark Haddad and some colleagues.
Yes, Rio. Sorry to interrupt. Shinichi Tatsumi.
Yeah.
So Ryosuke, who's one of the middle authors on that paper, I had met at a conference in Japan before the apocalypse. And he and I really got talking. And so he's a co-author on the first chapter I put in here. And so then in our understanding, their method is similar to what I'm putting up on the graph here with the price equation, but their methods have focused on different diversity indices than we have. I focused on Shannon entropy, and they've done stuff instead on Bray-Curtis in one paper, and then Jacquard similarity in another.
Yep, cool, thanks.
Thank you very much. Are there any other questions?
If there aren't, I will just, it's always useful to put
there will be some soon, but
oh yeah, oh, there are questions. Oh, that's nice.
yeah. Isadora,
okay.
Please go ahead.
Hi.
Hi.
How do you think the interspecific trait variation in these species are going to affect these dynamics of rarity and abundance and measures of diversity?
Okay, great. So I'm just saying this very slowly because it's early in the morning. So interspecific variation with among species.
No, yeah, it's ITV. It's the difference between the individuals in the same species.
Individuals within a same species.
Yeah.
That is a great question, and I have not grappled with that yet. So I'm going to go to I just wanted to share the screen again. So I've got this here. Okay. I don't know the answer to that, but I think we could start thinking through what would happen. So here, for example, we have competitive dynamics in the system. And this is in sort of like a classic Lotka-Volterra model where you're just keeping track of sort of the averages across two species and you put them on the phase portrait. I have not worked enough with intraspecific trait variation to think about it, but if you can put that on a phase portrait, you can then just recolor the phase portrait to see that in the case of a two species community. And so if someone would like to chime in, my understanding is that intraspecific trait variation, its effect depends. So like sometimes presumably it leads to niche differentiation, which makes competition weaker among species. What that will do in this case is sort of pull the trajectory up here. So on this, here's the carrying capacity of the two species when they don't interact. When they do interact, they're down here. So then if you put in trait variation within species to weaken competition, that moves you up this way.
Oh, nice. Yeah.
So I can tell you how to, yeah.
Sorry, kids can talk, sorry.
I was mostly just going to say that's most of what I know, but if you can put it on a phase portrait, you can do it. And if you can't, you can take the tools that are in that paper to convert from a species level evolutionary model, so ecological model, and then put it into diversity. I'm just naively going to assume that can be done, although I haven't done that myself for a model with intraspecific trait variation.
Oh, very nice. Yeah, because some species, they vary a lot. Like some populations are very not variable in some places, and they're very variable in other ones. And sometimes it's like correlated with some variable. So this could change this whole dynamics stuff. So it would be nice to take a look at that.
Yeah. If I remember right, so Dan Bolnick and I were chit-chatting about Jensen's inequality, and he had like sort of a review paper on intraspecific trait variation that he published a couple of years ago. Yeah. And I skimmed that and went, oh, that looks good. I should go back and read it. And I'm pretty sure that those pieces, if you squinted, could be put into this framework. And I totally agree it's important, but I have not done it. So I guess I'll leave it as an exercise to the reader.
Okay, nice. Thank you.
Thank you so much. Chris, please go ahead.
Yeah, thanks, Will, for the awesome talk. It seems like, the ecological concept space is always very fragmented, and, all these biodiversity, measures like Shannon entropy and hill numbers, they're always some kind of like island off the shore. I'm like, What's that
Yeah.
about? I don't know. So it seems like, so I really appreciate that you're building this bridge from that island to, like, you know, my mainland of, you know, dynamic models in ecology. So my question now is that since you built that bridge and you've shown that different kinds of species interactions and so on can lead to identical changes in diversity, what's your take on these diversity measures? Are they actually useful, or maybe we should just be like, we shouldn't use them because they're not particularly informative?
Yeah. Okay, so I am a geek on diversity measures. I think that they are useful, and I think that there's a lot that we can learn by doing them. But I think this is an interesting point of controversy and disagreement that's worth thinking about. So I'm just going to try to organize my thoughts. So one thing I'm going to say is that reading this paper convinced me that really clarified to me what we're missing when we look at diversity measures. In particular, like the ideas I'm borrowing are from evolutionary theory. And in evolutionary theory, there's a separation of timescales where sort of the changes in relative abundance happen on a different time scale as the changes in absolute abundance. And you can even see that in this graph here. So like this part of, there's a kink in the graph, and that's the separation of time scales. So in the first part of the graph, absolute abundances decline a lot. And in the second part of the graph, relative abundances change. So that's the ecological, that's the evolutionary theory version of sort of like why diversity is cool. It's because like relative abundances matter a lot. And that doesn't apply generally in ecology. It applies for interactions that are competitive and verging on neutral. And other dynamics are different, and we haven't really explored like how different that is. So that's sort of the big caveat, that like you are missing things when you're measuring diversity. But diversity, things that look like diversity are important in lots of different fields because they're a natural way to think about what we don't know. And I mean, I guess sort of that's what I put in you know, my one like blank part of the talk. So like Shannon entropy pops up in lots of different places, and there's good reasons for that. It's just this really handy way to think about uncertainty. And you know, when we think about to me, I think the uncertainty in which species are out there is an integral and important part of ecology. It's just not as complete as is a traditional model of community assembly, which focuses on absolute abundances of all species. But for argument's sake, that is not as complete as a consumer resource dynamics model, which includes information on resources of all the species, which is then not as complete as sort of like an even more reductionistic model.
Yeah, thanks for the thoughts. Yeah, I want to say I really appreciated your intuitive explanation of the Shannon entropy. That was awesome.
That's great. I might mention too, so I'm really pleased with that. Some of this I took from a paper by Steve Frank, which is called Natural Selection Part 5, How to Read the Fundamental Equations of Evolution in the Light of Information Theory, something like that, which is great. I'm glad I could share that interpretation, but that one has really been helpful for me and might be clearer than me, at least at 7 o'clock in the morning.
Thank you very much. Are there any other questions?
If there aren't, I'll put my contact information back up again. If anyone wants to get in touch, I'd be more than happy to do that. As I said, I'm really pleased to have a chance to share these ideas with other people in other parts of the world. It's really good to chat.
Absolutely, and thank you so much again for the talk. And thank you everybody else for coming as well. I guess then this is the end of the talk. We'll be in touch about the next seminar when that will be. And until then, we'll see you next time. So thank you again and see you next time. Bye, everyone.